All right, so in chapter 10, we're going to talk about muscle tissue. And in this chapter, we're going to talk about some of the major properties of muscle tissue, characteristics of muscle tissue, like the microscopic level and the gross anatomical level, as well as how skeletal muscles contract or produce force. We'll talk about the different types of skeletal muscle fibers, how they're organized, talk a little bit about exercise, as well as how muscles are named. And then a little bit on cardiac and smooth muscle. However, cardiac muscle is only found in the heart. Smooth muscle is found in hollow organs. That's more like AMP2 material. Okay, so most of the skeletal tissue chapter here is all about skeletal muscle. We'll get to a little bit on smooth and cardiac muscle. So just as an introduction to skeletal muscles, or just muscle tissue in general, we got three main types of muscle tissue. We got skeletal, cardiac, and smooth. Skeletal and cardiac are striated, so microscopically they look like they have striations. Smooth muscle doesn't have striations, and in the human body there's over 700 different skeletal muscles. We won't be going over 700 in lab. We're just going over 699. No, I'm just kidding. We're going over the big ones, the major ones, and it's far less than 700. Now, in terms of properties of muscle tissue, all muscle tissues are excitable, right? Which means they can respond to stimuli, like they can be excited to contract. So, for instance, if you want to, flex your wrists, well, then you have nerve endings that can actually communicate with the muscles of your forearm, causes those muscles to contract, and they're excitable, and then they can have contractility. So, contractility refers to the fact that muscle tissue doesn't necessarily just respond, but it also contracts in response to being excited. Okay, and contractility refers to the fact that muscles will shorten. Okay. So muscles will shorten, and when they shorten, they actually pull on something like a ligament, like a tendon, and that tendon can pull on skin or bone or some other tissues. Muscles are also elastic; they got to be stretched and then recoil back to a resting shape. They also need to be extensible, or basically need to be able to be stretched. So let's say, like, if you're going to perform surgery on somebody, which of these properties would you want to block? If you don't want someone to move around during surgery. Yeah, excitability could be one of them, right? If you prevent muscles from being excited, then they won't respond to any stimuli and they won't produce force, right? Or you can block contraction, right? Like you can block contractility and just prevent those muscles from shortening in response to being excited, if that makes sense. Now we know that muscles need to be elastic in the sense that they can recoil back to a resting shape. If you flex your arm, you don't have to physically pull your arm back into a resting position, right? It can just sort of relax back into rest. And part of that is due to the fact that muscles are elastic, and extensible just means stretchability. So we're going to talk about the characteristics of skeletal muscle tissue first, and then get into cardiac versus smooth muscle later. Now, each skeletal muscle is considered an organ because it contains multiple tissue types, right? So we've already talked in chapter 4 about the different types of tissue, but when multiple tissues come together, they form an organ, okay? So that means that skeletal muscle is made of more than one tissue. It's not just skeletal muscle tissue. It also includes blood vessels, other connective tissues, nervous tissue. You find all of that within a skeletal muscle, so we call skeletal muscle an organ because it contains these different tissue types. Skeletal muscle is striated, so it has a sort of striped pattern appearance to it, and it's usually attached to bones. Now, in terms of just the general functions of skeletal muscle tissue, it's involved in moving your body around. That makes sense, right? If it's attached to bones, it's going to move your body around in different ways. It helps maintain posture. It also helps it's involved with temperature regulation. So if you're cold, you'll shiver, and by shivering, you can raise your body temperature back up. It's also involved with storing and moving different materials, as well as supporting your body structure. So what we're going to do first, you guys, is talk about the organization of skeletal muscle at the microscopic level, and then kind of work our way up to the gross anatomical level. Now, each muscle is comprised of lots of different muscle fibers, and each individual muscle fiber is one skeletal muscle cell. So when we talk about like a muscle fiber, that's a cell, okay? Now, these things are microscopic, though. You won't see a skeletal muscle fiber or cell with the unaided eye, right? But if you look at muscles, they kind of appear to be fibrous, right? Like if you cut through a piece of steak or chicken or something, right? If you look at the cross section, you can see a lot of little endings. They look like fibers. You're not looking at the skeletal muscle cells because those cells are microscopic. What you're looking at are the bundles of cells. So skeletal muscle fibers are arranged in bundles called fascicles. And a fascicle is just a big bundle of skeletal muscle fibers or cells. Okay? These bundles are large enough to see with the unaided eye. Like if you look at the striations of muscle, or they talk about like, you know, when you cut up some meat or something, you're going to cut with the grain, where you're cutting with the fascicles, right? You're cutting along the length of those fibers, and those fibers are arranged into fascicles, which are basically just big bundles of muscle fibers. Now, each muscle fiber, which is a muscle cell, right? Each muscle cell or muscle fiber, same thing. Those on the inside of the cell have lots of what we call myofibrils. And a myofibril is basically a bundle of protein. So within these muscle cells, you find other bundles of protein tubes. And these protein tubes are the things that produce contraction. So it's the myofibril that you find within muscle cells or muscle fibers that are shortening during a contraction. So the entire cell shortens, and that produces force or tension, right? Now, these myofibrils are comprised of myofilaments. And these myofilaments are protein filaments that make up that bundle of protein fibers. And these filaments pull across each other, okay? So when they pull across each other, the whole myofibril shortens, which causes the entire muscle cell to shorten, and that's what produces sort of the contraction force here. Now, I remember looking at this as a student and thinking it looked kind of confusing because it's like tubes within tubes, right? So you just got to pick a starting point, and we'll talk about this. Could you hit the light for us? Thank you. So if we look at this picture here, you guys, what we see is skeletal muscle. And we can tell it's skeletal muscle. For one, it's attached to bone, right? And you can see that we have a tendon here, and that tendon eventually leads into our skeletal muscle tissue, right? Now, this is the entire muscle cut in cross-section here. So this would be like your biceps brachii, which is right in your forearm. And if you cut it in cross-section, what you'll find then is that within this muscle, there's big bundles of muscle cells called fascicles. So each one of these is a fascicle. Like here's a fascicle. That's a fascicle. Okay. Here's another fascicle. And each of these fascicles are surrounded by a type of tissue called paramecium. Paramecium is like a dense, irregular connective tissue that surrounds these skeletal muscle cells to form a bundle, which we call a fascicle. So the way the fascicle is all packaged together is it's surrounded by paramecium. And then deep to this fascicle, all of these little circles, in fact, here's a fascicle kind of pulled out for you guys right here. So here's a muscle fascicle surrounded by paramecium. Within this fascicle, we have lots of individual muscle cells, also called muscle fibers. Muscle fibers are a muscle cell. Okay, so all of these are muscle fibers or muscle cells. Now, they're microscopically small. You can't see them. You can't see them with the unaided eye. But if you look at this muscle fiber or muscle cell, we see it's got a plasma membrane, like all cells. And then within this muscle cell, look at this. We got more tubes. Okay, but these aren't cells anymore. You're not going to have a cell within a cell. What you're looking at now is actually a big old bundle of protein, which we call the myofibril. So the myofibrils are big bundles of protein within muscle cells, and these are the things that produce contraction. And if we zoom in on a muscle cell, so you can see our plasma membrane here, there are myofibrils within that. And you guys see they're kind of striped, like there's dark red, then kind of pink, dark red, pink. The striping pattern on the myofibril is what gives muscle tissue, skeletal muscle tissue, those striations. So when you look at skeletal muscle under a microscope, it looks striated. It's got stripes, right? Well, the stripes you're seeing are actually things that are sort of light. It's light that's bouncing back out of the cell, if that makes sense. So you're looking through the cell, and you're seeing the arrangement of protein within a skeletal muscle cell. Okay, that's the myofibril. Let's back up one more time. So looking at out here, you guys, we have our entire skeletal muscle, right? Within a whole skeletal muscle, we got these big bundles of cells called fascicles. Good. A fascicle is just a big bundle of cells. This is the one you'd see with an unaided eye. Like if you're seeing fibers in a muscle, you're actually seeing the fascicle, not a muscle cell. So you got a fascicle. The tissue that surrounds each fascicle is the paramecium. And if you look at a fascicle here, that means that all of these within that are individual muscle cells, also called muscle fibers. Good. So here's one muscle fiber, or a muscle cell, right? And then within this muscle cell, we have big old protein tubes called myofibrils. Nice. All right, cool. I know it's like tubes within tubes, and it's like it can get kind of confusing. You just got to pick a starting point, right? So if you look out here, then you know this is the entire muscle. Then you know the next layer of tubes within that tube of muscle are the fascicles, right? And we know that if you look at a fascicle, then the next layer of tubes within that fascicle of a tube is muscle cell or fiber. And then the next layer of tubes within that tube is the myofibrils. Myofibrils. Good. And we'll zoom in on this myofibril here in a minute. Now, the entire muscle is also surrounded by tissue, you guys, called epimecium. That's what holds the muscle together. Like, if you all have ever like prepared meat, like especially chicken has this, where like if you like take the kind of whitish tissue away off the outside because it's kind of tough and hard to eat, right? So that is actually epimecium. You know, it's obviously chicken epimecium, yeah. But it's basically like a dense, irregular connective tissue that surrounds the outside of the entire muscle. So if you're scraping that away from the outside of the muscle you're preparing to eat, then that's actually epimecium, right? So let's zoom in on this. Oh, so actually this shows all the layers. So we got our muscle, and then within an entire muscle we got big bundles of cells called fascicles. And then within a fascicle we got bundles of muscle fibers or muscle cells, right? And then muscle fibers or cells have big old bundles of myofibrils. And then myofibrils, what if I told you guys this is great? It's like Inception, right? Yeah. The myofibril actually is made of other proteins within that called the myofilaments. And there's two types of myofilaments. We got the thick filament and the thin filament. Okay? So right here is a thick filament. Next to it, we have thin filaments. So the thick filament has a little myosin heads here. The thin filament has what we call actin. We'll come back to that. Now, remember we talked about how the tissues, the entire muscle is arranged into connective tissue components, right? So what was the name of the tissue that surrounds the entire outside of that muscle? Epimecium. Epimecium. And then surrounding the entire one fascicle is paramecium. Good. And there's individual muscle cells have endomecium around them. But endomecium is more of a loose areolar type of connective tissue just to nourish those muscle cells. So that's what this is saying here, you guys. We have endo, peri, epi, right? So epimecium is what surrounds the entire muscle. It's made of dense, irregular connective tissue, so it's tough. Paramecium is what surrounds individual fascicles. It's also dense, irregular connective tissue. And then endomecium is a loose areolar connective tissue to nourish individual muscle cells. Now, deep fascia is what surrounds each muscle and separates muscles from each other. And superficial fascia is what separates muscle from skin. So later on in AP2, when we start dissecting fetal pigs, you guys will find superficial and deep fascia. And so deep fascia is actually what separates muscles, you know, like two different muscles that are side by side. Okay. Superficial fascia is what helps anchor skin to muscle. And we know that those things are connected because if you move muscles around, skin will move with it for the most part. So that's superficial fascia. And if I could describe to you guys what it looks like, to me it looks like spider webs. Fascia does. So both deep and superficial fascia look like spider webs. And when you guys start dissecting on the fetal pigs later, and you start pulling tissues apart, you'll notice these kind of spider webby like strings that like kind of hold your tissues together. Okay. Superficial fascia is what helps anchor skin to bone, and then between muscle. I'm sorry, did I say skin to bone? I meant skin to muscle. And then between muscle, then you have deep fascia. So muscle attachments include things like tendons, and so by definition, a tendon is what helps connect muscle to bone. But a big old broad tendon, we can call it aponeurosis. I'll give you guys an example of an aponeurosis. There's one called the Gallia aponeurosis that goes over the top of your scalp. Like, can you guys move your eyebrows up and down, right? Yeah, exactly. What if I told you guys that the muscle that you use to move your eyebrows up is actually found on the back of your head near the occipital region? So there's a muscle back here by your occipital bone, and it has a big broad tendon that goes over the surface of your scalp and then inserts right above the skin of the eyebrows. So when this muscle contracts behind your head, it pulls on that tendon, which pulls on the skin above your eyebrows, and that's how your eyebrows can raise up.
You can kind of feel that.
Yeah, when you contract it. So if you move your eyebrows up and you hold it back there, you can feel the muscle contracting. So I know, it's not weird. So it's a big, broad tendon that goes over the surface of your skull. Another example of an aponeurosis, you guys, you might have heard of tensor fascia lata, or like your TFL, like your fascia latae. You've heard of that before? So there's a big old broad tendon over the lateral part of your thigh here. There's also a big broad tendon over the surface of your abdominal trunk. So right over your abdominal area, there's a big old aponeurosis there as well. But the kind of the more interesting one to me, at least, is the Gallia aponeurosis, which basically pulls on your eyebrows, so like raise your eyebrows up. Kind of cool. One of the ways I discovered this was that I went to a comedy show like while I was taking anatomy, and I was laughing so hard that like the back of my head hurt. So I looked it up. I'm like, why would the back of my head hurt from laughing? Well, then it turned out like it's actually that muscle was sore because I was laughing so hard that my eyebrows were all raised up the whole time, and that muscle got sore behind my head because it felt like felt like someone punched me back there or something. But that's not Gallia aponeurosis. So it's actually pretty cool. Now we have origins and insertions, which are basically the different attachments for muscle. We know muscles will attach in at least two different places. Okay. Now the origin is the place that a muscle attaches that does not move. The insertion is the movable part. So I'll give you guys an example. Like your deltoid on your shoulder here, its origin is like the spine of your scapula and the acromion, and as well as the lateral 2/3 of your clavicle. Okay, that's the origin. Now its insertion is the deltoid tuberosity of your humerus. And when this muscle contracts, it causes your arm to abduct. Okay? Now what moved was my arm, right, the humerus. So that means the insertion then was the movable part. The insertion would be then the deltoid tuberosity. That's the part that moved when the muscle contracted. The immovable part were the ones that didn't move, right? Or the origin is the ones that didn't move. So like the spine of your scapula, the acromion, and clavicle will be the origin. That's the part that didn't move when I contracted that muscle. If that makes sense, right? Or what about like for muscles of the forearm? They've got tendons that attach to your carpal bones. And when these muscles of the forearm contract, you can move your wrist like in a flexion or extension kind of way, right? So if these muscles contract, it'll pull on your carpals to cause you to flex your wrists. So what's the insertion then for forearm muscles? If it causes your wrist to flex. Yeah, carpal bones, exactly. And the origin would be something like, the radius or ulna, right? Yeah, it makes sense. So the origin is the immovable part, and the insertion is the point of attachment that does move when the muscle contracts. So, this is an example here. So this shows the origin of biceps brachii, which is basically the supraglenoid tubercle of the scapula, as well as the coracoid process of the scapula. Now the muscle connects to the radial tuberosity of the radius. So when this muscle shortens or contracts, it causes your whole forearm to move up, right? So if the forearm is the movable part, this is the insertion. And if the scapula doesn't move while the biceps contracts, then this is the origin, if that makes sense.
So just the whole muscle moves besides the origin or just the little portion where the insertion is?
So the way that the way your forearm would move up then is that this entire muscle actually shortens. And as it shortens, it pulls. And as it pulls, then your whole forearm will move up with it. So just keep in mind that the origin is the part that doesn't move, and the insertion is the part that does move when the muscle contracts. So if we look at an individual skeletal muscle cell, we'll find that it's made of sarcolemma, sarcoplasm, and sarcoplasmic reticulum. Okay, that's weird, right? A lot of sarco. Well, sarco is the root for muscle or flesh, but in this example, we're going to say muscle. So sarcolemma is the muscle cell membrane, the specialized type of plasma membrane you find on skeletal muscle cells. Sarcoplasm sounds like cytoplasm, right? But sarcoplasm is the cytoplasm of skeletal muscle. And then sarcoplasmic reticulum sounds like endoplasmic reticulum, but this is a specialized type of endoplasmic reticulum you find in skeletal muscle. Okay, so We also have some unique features of skeletal muscles like transverse tubules, also called T-tubules. Basically, these transverse tubules are like canals that go deep into the muscle cell. I'll give you guys a good example of this. Y'all ever have like ant farms when you were a kid?
Yeah.
You can like order them online, and they even ship you the ants. They send you a little box of ants, and you can like pour your ants in there. My wife was saying that like she always got that for Christmas, but when she ordered the ants, you know, because it was Denver and it was so cold here, that the ants would like die in transit. So she never got like live ants to get like a real ant farm to work. I know, right? I know, right? I'm like, I can't wait to set my ant farm, right? It's like you get your ants, they're all dead. It's like an early introduction to just life, right? So yeah, exactly. Transverse tubules, you guys, T-tubules. They're basically just big tubes that go through the muscle cell. The reason why I said ant farm is that if you look at these things in cross-section, it looks like little canals that ants would dig, but through the muscle cell, okay? Now, what's interesting, though, is that these canals are basically just infoldings of plasma membrane that go deep into the muscle cell, but they have a closed end. Like the ends don't open up into the cell. It's just a big old fold that goes deep into the cell, and it forms a tube of that fold, if that makes sense. We call this a transverse tubule, and the purpose of this is to actually transmit electrical impulses deep into the muscle cell, because electrical impulses are necessary for the muscle cell to contract. Now, near the end of this transverse tubule, we have what are called terminal cisternae. Cistern is like a place to store something, right? And terminal means the end. So the terminal cisternae is basically a blind sac of Sarcoplasmic Reticulum that you find right by a T-tubule, and they form something called a triad, which is where the T-tubule interacts with the Sarcoplasmic Reticulum. So looking at this in sort of like an artist's rendition of the muscle cell, the name for the specialized membrane of a skeletal muscle cell is called Sarcolemma. So here's the Sarcolemma, which is basically the plasma membrane of that muscle cell. The sarcoplasm is all of this inside of the muscle cell, like cytoplasm, right? And then we have these green things over here. So these green things are the transverse tubules, and you'll see that they're basically like little tubes that go deep into the muscle cell, and they start spanning throughout the muscle cell, wrapping around these myofibrils, right? And the purpose of this is these myofibrils are the protein fibers inside of muscle cells that are involved with contraction. So it makes sense then you want these transverse tubules then to wrap around each myofibril to help stimulate a contraction to begin, right? Now, something else that's associated with this myofibril is the Sarcoplasmic Reticulum, or SR. So Sarcoplasmic Reticulum is like endoplasmic reticulum, and it's basically full of calcium. So think of the Sarcoplasmic Reticulum as just big old bags of calcium. And the purpose of calcium here is to help initiate a skeletal muscle contraction. And we'll come back to this later. But what happens is, if you notice that we have our Sarcoplasmic Reticulum associated with transverse tubules, so what happens is when electrical impulses travel through the transverse tubule, or T-tubule, when it gets to the Sarcoplasmic Reticulum, it can stimulate that SR to release calcium into the cell. And it's calcium in the muscle cell that actually stimulates a contraction to begin. But we'll get back to that mechanism here in a little bit. Just kind of keep that in mind, right? So here's the weird part, you guys, is that when a nerve fiber excites this muscle cell, it stimulates electrical impulses that travel along the length of the sarcolemma, right? So they go across the sarcolemma, it goes down the transverse tubule, deep into the muscle cell, where that electrical impulse then can start to stimulate Sarcoplasmic Reticulum to release calcium. You got it. And that calcium, once in the sarcoplasm, can diffuse throughout the muscle cell, and calcium is the stimulus to get muscles to contract. Okay? So the contractile units of the muscle are the myofibrils. So the myofibril is this cylindrical structure that extends the entire length of the muscle cell. So when we talk about muscle protein, this is what they're talking about, you guys. Myofibrils are muscle protein. So if you hear about like someone has muscle protein wasting or atrophy, or muscle protein hypertrophy, right, where muscles get larger, we're talking about increasing or decreasing the number of myofibrils in that muscle cell. And these myofibrils have the ability to shorten. And as they shorten, they produce tension because they're pulling on the cell itself, right? So these things that you find inside the muscle cell, let's say my arms are the myofibril, right? When the muscle contracts, then the myofibrils can shorten, but because they're attached to either end of the cell, it makes the entire cell shorten with it. But that muscle cell is attached to tendons, right? And those tendons are attached to bone or skin. So when the muscle cell shortens, it pulls on tendons that pull on something else, right? And they could pull on skin, they could pull on bone, other muscles. And the myofibrils are the things that are causing the muscle cell to shorten. So these myofibrils have the ability to shorten. What I mean by shorten, you guys, I don't mean just like collapsing. They're actually pulling as they get shorter. And they're pulling because these filaments you find inside the myofibrils are pulling against each other. And when they pull against each other, it causes the entire myofibril to shorten with it, which then causes the entire muscle cell to shorten with it, which then pulls on a tendon, and that can help move your arm to flex. That's kind of cool. This is occurring at the cellular level. Now, myofilaments are the things that make up myofibrils, and there's two varieties. We've got thick filaments and thin filaments. The thick filament is, like it sounds, guys, thicker, and it's associated with what we call myosin heads, the little fists, okay? And the thin filament is thinner in appearance, and it has attachment sites for the myosin heads. So the myosin heads on a thick filament attach to the thin filament, and when they attach, they can pull on it. So it pulls on that whole thin filament in a particular direction. And this forms something what we call the sliding filament theory of muscle contraction. So remember, the thick filaments are the thickest. They're 11 nanometers in diameter, which is actually pretty thin in terms of our scale of understanding, but it's thicker of the two. So they're about twice as thick as the thin filaments. They're composed of bundles of myosin, and myosin is an enzyme that can use ATP to contract. So let's say if my whole arm here is myosin and my fist is the myosin head, I can use ATP then to start doing this. Okay? So if this is the myosin on a thick filament, what if I told you guys that myosin on a thick filament just does a whole lot of this? And when it reaches up, it grabs onto the thin filament, contracts, unbinds, recocks, rebinds, contracts, unbinds, recocks, rebinds, contracts, and it keeps doing this over and over and over. But what's happening to the thin filaments is they're getting pulled on. And as they get pulled on, they're moving more and more centrally, which is causing the entire myofibril to shorten with it. Okay? Now, what this means then is that it requires a tremendous amount of ATP to get your muscles to contract, because those myosin heads use ATP to do that motion. Like they don't just move like this on their own. They need ATP to start moving, and they use the chemical energy in ATP to make kinetic energy, or basically movement. Now the thin filament is comprised of actin, which is the binding site for myosin, troponin, and tropomyosin. We'll come back to this in a little bit, guys. Now, we have these actin subunits, which is what the myosin heads bind to. But what if I told you guys that these actin binding sites aren't always available to be bound, right? If they were, then our muscles would always be contracted. So what happens then is that there's something that blocks the way while your muscles are relaxed. So while your muscles are relaxed, we have tropomyosin and troponin that help block the binding site for actin. Okay, let's check this out. So if we look at the myofibril up close. By the way, guys, we've zoomed in on the myofibril here. So here's our skeletal muscle cell, like we saw. Here's the myofibril within that skeletal muscle cell. And if we zoom in on that myofibril within the cell, here's what it would look like. Very microscopically. Like you need an electron microscope to see this kind of detail. Now, what we see here is the thick filament, because it even looks thicker, right? It's associated with thin filaments nearby. But what is on the thick filament? What are these little fist-looking structures? Myosin heads, good. And myosin uses ATP to contract, right? So these things can use ATP to start kind of wiggling. They kind of wiggle around. They start doing a lot of this, okay? They're using ATP to do that. Now, if they can bind to something on the thin filament, then they can start to grab on. They can form a cross bridge, and they can start to pull on these thin filaments towards the M line or center line here, right? But because this is all linked up in a chain within a myofibril, the entire myofibril will start to shorten when myosin heads start pulling on the thin filament, okay? Now, if we zoom in on the thin filament, what we see then is we got our little actin subunits. What's the function of actin again? Nice, very good. It's a binding site for the myosin heads. And then look at this, you guys. We got this little green ribbon looking thing going across that binding site. That one's called tropomyosin. Tropomyosin is a blocking protein. It blocks the binding site while your muscle's relaxed. Okay? Because if the myosin heads can't bind to the binding site on actin, then you won't produce contraction. So the muscles relax there. Okay? And then what's associated with tropomyosin then is troponin. So this little blue thing here is troponin. And troponin is a calcium-sensitive protein. So that when calcium binds to troponin, troponin pulls on tropomyosin, moves tropomyosin out of the way, and then that exposes the binding sites on actin. Okay? So let's back this up. What was the function of the myosin head again? To contract, right? But it only will contract if it binds to actin on the thin filaments. Nice. But what blocks the binding site while the muscle's relaxed? Tropomyosin, which is that green filament looking thing. Now, troponin, what's the function of troponin?
It moves the tropomyosin out of the way so that you can bind.
Good. And it also responds to an atom. What atom? Calcium. You got it. So troponin is a calcium-sensitive protein. When calcium binds to troponin, troponin changes shape. And while it changes shape, it pulls on tropomyosin, moves tropomyosin out of the way, and then that exposes the binding site now in the thin filament. So now what can the myosin heads do? You got it. They can bind actin. Then they can use ATP to start pulling on actin. They can unbind, recock, rebind, recock, unbind, recock. And then the whole thing starts to shorten up. And this is occurring like instantaneously, pretty much. Huh?
So it sounds like it would take like a lot longer.
Doesn't it? I know, yeah. All these steps,
Like
right?
we could all just move like really slow.
Yeah, it's like, yeah, whoa! That's funny. Yeah, if this occurs at our scale of understanding, basically instantaneously. You're right. But it's pretty cool. So what this is showing you guys is just a summary of those different parts of muscle we talked about. So what was the sarcolemma again? Plasma membrane of the muscle fiber, or muscle cell, right? What sarcoplasm was the cytoplasm of the muscle cell. How about sarcoplasmic reticulum? It's the
equivalent of the smoothie R.
good. It's the equivalent of smoothie R, but it's full of calcium. Ah, we're kind of tying things together here, right? So it's full of calcium. And then its sarcoplasmic reticulum is associated with T tubules at a place called the transverse, or the terminal cisternae. What was a T tubule like? Kind of like an ant farm, right? But like, what is it? It's mostly just an infolding of membrane deep into the muscle cell. What was the function of a T tubule?
To transmit electrical impulses.
To transmit electrical impulses. Good. And what about the myofibril? What's that essentially?
That's the,
It's inside the muscle cell, and the myofibril is basically a big old bundle of protein. Good. Now there's two types of protein that make up the myofibril. We got thick filaments and thin filaments. What's on the thick filament?
the myosin heads.
Nice, guys. Myosin heads. Good. And the thin filament has three different proteins. We got actin, tropomyosin, troponin. What's the function of actin?
The binding site.
Binding site for
Myosin.
myosin. Nice. And then what's the function of troponin?
It blocks the binding site.
Well,
The troponin.
Yeah,
is
troponin.
It moves the tropomyosin out of the way.
Good. It interacts with calcium when calcium's present, right? Calcium's got to be there to bind to troponin. But when calcium binds to troponin, troponin then pulls on tropomyosin to move tropomyosin out of the binding site for actin, and therefore the binding site is now exposed for the myosin heads to bind and then contract to produce some force. We also have another protein here called connectin, you guys. It's basically a big filament of giant protein, and it helps provide passive tension within your muscle. And then nebulin, which is also a big giant protein, it also helps regulate the length of the thin filament. So there's other proteins here involved.